Bismillahirrahmanirrahim. 9700 November 24. Question paper 1 2. First video, question 1 to question 20. Question number 1. A light microscope is used to observe two structures that are 200 nanometers apart on the slide. What is the actual distance between the two structures when the magnification is changed from 40 to 400? You see, the actual structures are 200 nanometers apart. Whether you magnify it will of course then look as if it's far apart, but actually it says what is the actual distance. The actual distance would always be the same as it was. I mean, if the size of your iPhone says 5 inches and you make the picture of it bigger, but the actual size will remain the same. It is the size of your iPhone will remain the same even if the picture is very, very highly magnified. You have magnified your iPhone or your phone or your mobile or a sharpener or a pencil, but actually it's going to remain the same. So that's why the answer is C to this. I said, okay, a catch in English. It isn't doing anything different. It's just a catch in English that they're trying to see if you understand the English behind it. So question number 1, the answer is C. Then question 2, a cell is shown in the micrograph. It's a micrograph. Which statement explains how it is possible to identify the type of microscope used to produce the micrograph? The nucleus is visible, so an electron microscope was used. The endoplasmic reticulum is not visible, so a light microscope was used. That's wrong. Endoplasmic reticulum cannot be seen in a light microscope. Chloroplasts are visible? No. So the answer is D. Ribosomes are visible, so an electron microscope was used. We cannot see ribosomes. They're too small to be seen by a light microscope. So this was wrong. And the answer is D. Then coming to question number 3. Which statement supports the fact that mature plant cells contain organelles that carry out the same role as lysosomes? What do lysosomes contain? Hydrolytic enzymes. So the answer is A. A range of hydrolytic enzymes can be found within mature plant vacuoles. Glycogen is not found in plant cells. Double membrane bound vesicles are formed from, of course not, Golgi vesicles are single membrane bound vesicles. Vesicle formed from the cell surface membrane contains enzymes. No, cell surface membrane vesicles are not there. Lysosomes are Golgi vesicles. Question number 4. Which cell structures contain cisternae? Chloroplast, no. Thylakoids, mitochondria has cristae, not cisternae. So the answer is C. Endoplasmic reticulum and Golgi body have cisternae in it. Then question number 5. The diagram shows some cell structures of one type of cell. Which labeled cell structure are present in a typical eukaryote and a typical bacterial cell? So you got to understand it's 1, 3, 4, and 5. 2 is the cell wall, which is not present in typical eukaryote. Eukaryotes are plant and animal cells. So cell walls, and this is also made of a peptidoglycan. So which labeled cell structures? So it has to be, uh, that's why the answer to this is B. 1, 3, 4, and 5. What was 1? 1 is cell membrane. 3 is circular DNA, which is found in eukaryotes in mitochondria and chloroplasts. And 4 is ribosomes. And this is mostly 70S ribosome because this looks like a bacteria. And so 70S are found in mitochondria and chloroplasts. And 5 is the cytoplasm. Question number 6. The diagram shows part of a collagen fibril made of collagen triple helices. The collagen triple helices are linked to each other by one type of bond. The bond is labeled as X in the diagram. So that has to be a covalent bond. That's something you have to know. There's no question of understanding it or anything. They're just covalent bonds given in your book as well. So covalent bonds joining the units of the three polypeptides twisted around each other, and then you can see the staggered ends, and then you can see them joined by these covalent bonds. Question 7. The table shows some information about the polypeptides that make up hemoglobin. Total number of amino acid residues in polypeptide chain alpha globin is 141 and beta globin is 146. And position of amino acid cysteine position is 104. Position means 1, 2, 3, 4. And in the beta globin, the position of cysteine is at 93 and 112. Now, scientists study the region of the beta globin polypeptide chain containing the amino acid cysteine at position 93. They found that the region faces outwards when no oxygen is attached to the heme group. This region faces inwards when oxygen is attached to the heme group. Replacing cysteine with a different amino acid reduces the Bohr effect. What can be concluded from the information about cysteine in the hemoglobin? More than 1% of the amino acids in one hemoglobin protein are cysteine. That's the correct answer. But then, why are the others wrong? In alpha globin, there is cysteine closer to the end of the polypeptide. Alpha globin 141, it was 104 with an unreacted card. That doesn't we don't get that information from the question. The replacement of the cysteine at position 93 decreases the affinity. No information, no information. No, we don't get this from there, and this one is wrong as well. Question number 8. Which features of glycogen distinguish it from starch? Remember, glycogen, starch has two types, amylose and amylopectin. Glycogen has only one type, 1,4 and 1,6 bonds. All glycogen molecules are highly branched. Yeah, that's the answer. But then all glycogen molecules are polysaccharides. We're saying which feature of glycogen distinguishes it from starch. I mostly say, this checks your English rather than biology. All glycogen molecules contain alpha glucose, but starch also contains alpha glucose. Only cellulose contains beta glucose. All glycogen molecules contain 1,4. Starch also contains 1,4 glycogen. It says distinguishes it from starch. Starch, amylopectin is not highly branched. It is branched, but it's not highly branched. So that's why the question number 8 is A. Then coming to question number 9, the diagram shows a biological molecule. Which molecules would be produced if the biological molecule was hydrolyzed? I mean, this is a triglyceride. So it would be what? Fatty acids. It says if it is hydrolyzed, we need water to it. If you add water, then only it will break up into its constituents. So it will break up into fatty acids and glycerol. That's why the answer is C. You have to need water to, you need water to break these bonds. You need 3 water molecules to break this bond. Question number 10. A mixture of glucose and starch solution was placed in a length of dialysis tubing and the tubing sealed. The tubing was thus placed in a boiling tube containing distilled water. So we've got this as the distilled water here outside it. 2 samples were immediately removed from this water 0 minutes and tested with either iodine solution or Benedict solution. This was repeated at 10-minute intervals for 30 minutes. The iodine solution gave an orange-brown color each time. So that shows that the starch could not come out of it. The starch remained inside. So the starch remained inside and could not come out of the dialysis tubing was a large molecule. The table shows the Benedict's test. You see the glucose would have come out and that would have given a positive. So that's why it was blue in the beginning, then became green, yellow, and red, because more and more glucose diffused out. More and more glucose diffused out into the water. What may be concluded from these results? The pores in the whisking tubing are too small for a starch molecule to pass through. Yes, that's correct. Glucose diffuses through the whisking tubing down a diffusion gradient. Water diffuses into the whisking tubing. Water diffuses, so what is happening? The starch remains, the starch doesn't come out, but the glucose is increasing. That's why it gave you a green color, then a yellow, and then a red color. So that is why the answer is B. 1 and 2 only. Water has nothing to do with this. Why would water diffuse in? Glucose is moving out, so it's becoming a concentrated solution. So nothing to do with what may be concluded from these results. These results. So the answer to question 10 is B. Then coming to question number 11. Which molecules are globular proteins? Amylase is an enzyme. Hemoglobin is a globular protein. DNA polymerase is an enzyme, globular protein. So question 11, the answer is A. All these three enzymes, hemoglobin, an enzyme again, DNA polymerase is an enzyme, all are globular proteins. Question number 12. The initial rate of reaction catalyzed by an enzyme was measured at various substrate concentrations. Which graph shows the effect of low concentration of non-competitive inhibitor on the reaction? So low concentration of non-competitive inhibitor. Now you can see this is without the inhibitor and this is with the inhibitor. So the answer is D. Why? Because as you can say, with the inhibitor, the rate of reaction has decreased considerably. With a non-competitive inhibitor, it never reaches the Vmax. So it has to decrease. So this was without the inhibitor. Was this one? So this is without the inhibitor. Gout is a type of arthritis in which small uric acid crystals form inside and around the joints. It causes sudden attacks of severe pain and swelling. The diagram shows how uric acid is formed from hypoxanthine catalyzed by the enzyme xanthine oxidase. So we've got hypoxanthine and then we've got xanthine, and the enzyme is xanthine oxidase here. So the active site of xanthine oxidase, the substrate hypoxanthine would fit into it, and then xanthine fits into it and then forms uric acid. Now gout can be treated using a drug allopurinol which has a similar shape to hypoxanthine. So it is a competitive inhibitor because it has the same shape as the substrate, hypoxanthine. What can be concluded from this information about how allopurinol prevents the formation of uric acid? It binds to the active site of xanthine oxidase instead of hypoxanthine, resulting in reduced production. Yes, that's the answer. Now, why is B wrong? It binds to another part of xanthine oxidase. This changes the shape of the active site, but the shape of it is the same as hypoxanthine. You can see it's the same as hypoxanthine. So this is wrong. Then it disrupts the hydrogen bonds with xanthine oxide, denatures the active site. Why would it do that? It has the same shape as hypoxanthine. It hydrolyzes the peptide bonds. Why would it hydrolyze the peptide bond? It's not an enzyme. So the answer is A. Now coming to, this is a very interesting question. It took me quite a while to really figure this out. Uh, Question number 14. Phospholipids are formed in a similar way to triglycerides. In a similar way to triglycerides, please remember, formed. A sample contains 6 phospholipid molecules. The molecular weight of phosphate ion is 95. The molecular weight of each individual fatty acid is 282. The molecular weight of glycerol is 92. The molecular weight of water is 18. Now please pause the video here and figure out what is the answer. The answer is B. But how is it B? Let's look at it. How is it B? You see, I did reverse maths in this. I really didn't understand this question, and I said, okay, how are we going to figure this out? So 6 phospholipid molecules would have 6 phosphates. So 95 into 6 is 570. Now 6 phospholipid molecules would have, because 2 fatty acid chains, so that we'll multiply 282 by 12, so 3384. And then we would have glycerol, we'd have 6, so 92 into 6. So 5 to 2, this comes 4506. But then in order to make it, because it says phospholipids are formed in a similar way to triglyceride, we would have 3 water molecules exiting. Each time the bond forms between glycerol and the fatty acid chains. So we need water to add the, so water would have to be minus from that. Because it says a sample contains 6 phospholipid molecules. So when the bond is formed, water exits. So 18 into 18 water molecules would leave. So 18 into 18 is 324. So 4506 minus 324. I'm sure there are other ways by which students would have handled this question. This was my way of handling the question. So question number 14 is B. Question number 15. Which statements about phospholipid in cell surface membranes are correct? Fatty acids allow most ions to pass through. No, it's a hydrophobic part. How can it allow ions to pass through it? Hydrophobic tail point inwards facing each other. That's right. They turned inwards facing each other. That's correct. All polar heads face the cytoplasm. No, this is polar and this is also polar. So polar is on the outside as well as on the inside towards the cytoplasm. So this is wrong. This was wrong. So the phospholipids help with the flexibility of the membrane. Yes, that's correct. So the answer is 2 and 4, which is D. Question 16. In an experiment, pieces of onion epidermis are put into 3 different concentrations of sucrose. Solutions P, Q, and R. The pieces of onion are left for an hour and then examined using the low power of a light microscope. Each diagram shows one cell from the epidermis that was placed in each of the sucrose concentrations. What explains the appearance of solutions in cell solution Q? The concentration of solution Q is equal to the concentration of solutes in the cell sap. The concentration of solution Q. So there should have been no net movement. But then you can see in P it's become fully turgid, in R it has plasmolized. The cytoplasm has the same concentration of sucrose as solution Q. That's not possible. Sucrose is not present in the cytoplasm. The water potential of the cytoplasm is equal to the water potential of the vacuole, but that doesn't cause the water to move in and out. So the answer is D. The water potential of the cell sap is equal to the water potential of cell Q. Question number 17. The electron micrograph shows rod-shaped bacteria. Length is 2 micrometer, diameter is 0.5 micrometer. The actual length of the bacteria is 2 and the diameter is 0.5. Assume that the bacteria is cylinder shaped. What is the surface area to volume ratio? It's 9:1. Please find out how do you calculate the surface area to volume ratio of a cylinder, and I'm sure you can figure it out. Next, coming into question number 18. Chickens have 78 chromosomes in the nucleus of a body cell. How many DNA molecules are there in a chicken body cell at the start of prophase of mitosis? So what should have happened? You see, S phase of interphase must have occurred. So it would have doubled. So that is why the answer is D, because one DNA molecule is this. This is one DNA molecule, ATGC. There are millions of these nucleotides. One chromosome, this is one DNA molecule. Now there would be another because you see there are 2 sister chromatids. These are sister chromatids. And this would have occurred, DNA replication would have occurred in S phase of interphase. So in the prophase, they would be double the number. 78 chromosome means 78 DNA molecules, but now there'll be another sister chromatid attached to it. So this would have 78. So this is one chromosome, and then this is the sister chromatid. So 78 chromosomes will be 156. The photomicrograph shows cells undergoing mitosis. Which statement describes what will happen next in cell X? Cell X is what? Prophase, metaphase, anaphase, telophase. In metaphase, what happens? Chromosomes line up along the equator of the cell and attach to the spindle. So that's why it's going to be B. Which statement describes what will happen next in cell X? This is prophase. So prophase, the next is metaphase. Interphase, prophase, metaphase, anaphase, and telophase. Question number 20. Which processes occur in bone marrow cells that are in a mitotic cell cycle? Phosphate groups bind to ADP molecules, form ATP. Bonds form between nucleotides in a DNA strand. Hydrogen bonds form between tRNA and mRNA. So the answer is A. All these things are occurring in the cell cycle. Because when organelles replicate, a lot of energy is needed, DNA replication is taking place, and then a lot of protein synthesis is taking place, ribosomes are being made, mitochondria, organelles are replicating, so mitotic cell cycle. Then question number 21. The enzyme telomerase prevents loss of telomeres after many mitotic cell cycles. Which cell needs to transcribe telomerase enzyme? Stem cells divide repeatedly, so they will be needing it, and activated memory B lymphocytes multiply rapidly, so they undergo mitosis, but this one will not. So that is why 21, the answer is B. 1 and 2 only, they will be needed to transcribe a lot of telomerase enzyme, because they're saying after many mitotic cell cycles. A polypeptide molecules contains the amino acid sequence glycine, leucine, lysine, valine. The table shows the DNA triplets for these amino acids. Now I've done that for you. You must do it in this order: DNA, mRNA, and tRNA. You usually get this wrong in the A's and the T's and the U's. If you don't practice this, of course you're going to get it wrong, definitely. So it's all your mistake. Don't blame the teachers, don't blame the school, don't blame tutors, don't blame anybody else, blame yourself. Because this is DNA, this is RNA. RNA does not have any T, it has U in it. 23. The diagram shows a section of a strand of DNA. Which type of bond is labeled X? This is between the nucleotides. It can be anything else but a phosphodiester bond. Question number 24. Which row correctly describes cytosine? What is cytosine? Cytosine has 3 hydrogen bonds. Adenine and guanine are double ring. This is a single ring. So single ring is not a purine, it's a pyrimidine. It's all factual. You've got to know it. You've got to remember it somehow. I remember it. Adenine and guanine are purines. It's got a U in it. It's a double bond. There's a U in double. So I remember it by making a mnemonic. I know we all have to remember a lot of bio is all nothing but rote learning. So the answer is C, ring structure single, 3, and it's a pyrimidine. Then we come on to question number 25. During the production of protein molecules, only one strand from the DNA double helix is used. You see, this is the double-stranded DNA, and then one of it is copied by the mRNA. And then it, of course, this unzips and then zip backs. Once it's copied, the mRNA leaves and it zips up again. Which name is given to the DNA strand that is used to produce a new protein? So the answer is C, template strand. Leading and lagging strands are in DNA replication. Non-transcribed strand would be this one. Which name is given to the DNA strand that is used to produce a new protein? Question number 26. The diagram shows a longitudinal section of a phloem sieve tube with a companion cell. Where are the mitochondria located for the release of energy for co-transport? Naturally, they would be in B. Why? Because you see, this is the companion cell. And in the companion cell, the mitochondria, large number of mitochondria in the companion cell for the co-transport. Why? Because H ions have to be pumped out and then H ions diffuse back and bring along with them sucrose. Question number 27. The diagram shows a phloem sieve tube element and a companion cell. You see, this would be the phloem sieve tube element because you've got these sieve plates here. And this would be the companion cell. Right? So this is the companion cell. Now it says that are involved in translocation of sucrose. Which process correctly describes the translocation of sucrose through these cells? Now the answer is B. Protons move from Y to Z. Yes, Y is pumped into the hydrogen ions are pumped, so protons move from Y to Z by active transport. Sucrose moves from Y to C. Sucrose moves into the companion cell. So this is wrong. Protons move from Z to Y by diffusion. No, that's wrong. Protons move from X to W. Rubbish. Question number 28. Which types of molecules are co-transported into companion cells? Monomers and disaccharides. Amino acid is a monomer. Sucrose is a disaccharide made up of glucose and fructose. Question 29. The diagram shows a section to the human heart. Which label is correct? C. Right atrium. This is something you've got to know or you just don't know it. There's nothing of no explanations to it. You just have to know this diagram. We continue with question number 30 to 40 in the next video. And please, thank you for subscribing. Thank you for watching. And please do let me know if there are any comments in which I can help you more in your studies for your A-level biology for the upcoming exam.